your ionization. They can interfere with your ionization. They can interfere with your, interfere with your ionization, with your protein assays, with just identification in general. But also getting genomic sequencing from soils is also challenging. There's a high complexity, there's a high microbial diversity. There's lack of sufficient databases for functional annotation. So you might get a ton of gene calls and not have any idea what they are. They come back as hypothetical, unknown, conserved hypothetical, which doesn't really help a lot when you're trying to figure out what function is going on in your system. And then even if it comes back in an annotation, annotations are based by other annotations. So if you don't have a really good annotation database, you're still struggling with what is the function. But we have started to overcome some of these challenges. The unknown database, the unknown proteins and functional annotations, I think is actually going to take a real effort by our funding agencies to address well. So I'm probably not going to talk about that. That's just something that we all have to deal with. Okay. Can you see my pointer if I actually, can you see my cursor? Thanks. So a soil metaproteomics workflow, at least in our hands, is we take a soil sample and we have different methods, and I just have direct and indirect extraction of proteins. And I'm going to talk a little bit about the way we can extract proteins. But we extract the proteins are digested to peptides. Now, at this point in time, the peptides can be analyzed directly by LCMS. We get our data, the data is then searched, and we use MSGF Plus, which is an open source searching algorithm to take our LCMS data and compare it back to a metagenome. There's other programs out there, Sequest, Mascot, I'm blanking on the rest of them, but MSGF is the one we decided to use because A, it's open source and we want to multiplex it across all computers, and Sequest started getting really expensive when we had 20 nodes going and we had to buy licenses for each node. Plus Sante Kim was in our, who developed this, was in our group for a while. So he actually helped us develop some further advancements to it. But then from our peptide and protein identifications, we can look into what the biology, the taxonomy, and the function of our microbial community is. So there are different ways of extracting extracting proteomes from samples. One of the ones that we like to use is called MPlex, and what it is it's a modified Folch extraction where you use methanol and chloroform to separate lipids, polar metabolites, and proteins so the polar metabolites go into the methanol layer, the lipids go into the chloroform layer, and the proteins go along this beautiful interface. And if you're going to try to do metabolomics and lipidomics from the same sample, this is a great method because you get all of them from the exact same sample. Now, I would like to say that soil separates like this and it's very beautiful and everything works well, but no, this is what it actually looks like when you do a soil sample. We're still getting polar metabolites, we're still getting lipids, we get this protein interface and we get a bunch of junk. So what we end up doing is we end up taking the methanol layer off and getting our metabolites. We end up getting our lipid layer off, or the chloroform layer and getting our lipids. And then we extract the proteins from this interface as well as the gunk at the bottom. Soils are messy. There's just a lot to deal and learn with them. And we have found that there's still a lot of proteins sitting in this bottom layer. However, MPlex is also it's a more complex method than just a direct extraction. So if you want to do multi-omics, you want to look at an MPlex method because it really does reduce sample need as well as you're extracting everything from the same sample. But if you're just wanting to do proteomics on your sample, there are other methods. And we have found that the MPlex extraction isn't always the best. And so this is just a comparison of 3 extractions, the MPlex, the SDS extraction, and a MoBio kit, which isn't available, I believe, anymore, but I think that other people are coming out with other different kits. And we get different proteins. Now, I'm going to say the protein yield here isn't high because we didn't do extensive running on these samples. When you're looking at wanting to run tests, and I recommend running a test of different extractions on your samples when you first get them, you run it through the mass spectrometer once, because as long as you're comparing one run to another run to another run, you don't need to take a lot of mass spectrometry time to get these numbers. And so the numbers aren't uber high because we did one run and we're just looking at how many proteins we got out. So in looking at this, the MoBio kit for these samples, and I'll explain it in the next slide where all these samples came from. The MoBio kit worked the best. Our SDX extraction worked okay. We actually have an indirect extraction that I don't have shown here, where we can pull the microbes off the soils and then do an extraction from them that actually works quite well as well. So again, it depends on what you're trying to get to. If you want to do the multiplex, you want to do MPlex. If you don't, you want to do proteomics, you might want to try a different method, and try different methods, because every soil I've ever worked with has been different. And every soil has had a method that works better than the rest. So the other thing that, if I'm going to go back really quickly, the other thing that you want to consider doing is this fractionation. So the peptide, when we do the peptide, when we do the tryptic digestion to get peptides, we're looking at potentially 50,000 peptides in a sample. You can imagine how many peptides we're going to get from that. And I get 50,000 because if you have a soil sample that has 100 microbes at an average of 5,000 genes per microbe, that's 50,000 proteins. We're never going to get that many, and you're never going to see them all, but it's a very complex sample. So if you just run that through once, you're going to get proteins, but you're going to get the most abundant proteins, because what happens is you end up with co-eluting peptides where you get charge suppression. So you don't see the lower abundance peptides, and because the higher ones take all the charge in the mass spectrometer. So what we've ended up doing is doing a high pH fractionation where we take the peptides and we separate it out using a high pH fractionation into 96 fractions. And then those 96 fractions are concatenated in what we call smart pooling. So if we have 96 fractions, if we want to bring it down to 6 fractions, we'll take 1, 7, 13 and combine those together, because then our salt concentration across all of our fractions is exactly the same. And we get the same sort of salt ion suppression that we would, and we do SPE, but you don't want your high, if you just do, I'm going to collect 1 through 6 and then 7 through 12, et cetera, you get a lot of salts at the end of our high, even at the end of the high pH fractionation, that interferes with your signal. If you do the smart pooling, you end up normalizing all the salt across all the fractions, and you're still getting all of your information. So going back to why do we want to do fractionation? Well, if you look at the sample the sample I'm showing here, if we do a one-dimensional run with a 2-hour separation with no fractionation, we got 8,884 peptides, which corresponded to about 1,000 proteins. If we do 12 fractions in our high pH fractionation and run them for 1-hour separations, and there's reasons why you'd want to do this, you get about 60,000 peptides and 11,000 proteins. And then if you run it for 2-hour separations, you get about double. We doubled the separation time. We get almost double the number of peptides, and again, almost double the number of proteins. Now, why do you want, you might, when we're running just these tests up here, we don't fractionate, which is why our numbers are relatively low. You want to do some sort of fractionation if you want to get any sort of biological information, but the number of fractions and the number and the separation time you use for the fractions comes at an opportunity cost. So if you want to do a lot of fractions with a long separation and get a lot of peptides, you're not going to be able to do as many proteins, because everybody has a limited amount of something. It could be money, it could be instrument time, it could be sample. So anything you do and every choice you make comes with an opportunity cost of not doing something else. So it's very crucial to find the balance of what your method is based on what your resources are. And that can sometimes be a tricky thing to do. So the next thing I'm going to talk about is the genome, because the genome is extremely important in how you, in the kind of data you get. And so in this case, our samples came from Hopland, California, and we were looking at what happens if we do a glucose amendment to the soils to see how the carbon is actually, the glucose amendment was a mimic of a plant exudate. We know that plants exude sugars. So we just tried this glucose because, you know, glucose is picked up by microbes pretty easily, just to see where the carbon goes and to take a look at what was going on. There's another reason that you'll understand later why we picked glucose, but so we added the glucose to the soil, and then we analyzed it using fractionation as was shown before, and looked across a number of genomes. Now we actually have 4 different genomes for this, but I only have 3 shown here, just to show you the different types of genomes. The first one is a comprehensive deep genome sequencing of everything. So we took the soil, we got a very deep sequencing, we collated all the data and annotated it, and we got about 30 million gene calls. And what this is really good for is function, because you don't have a lot of taxonomy information. The next thing we did is we took this comprehensive metagenome and we binned it into, using binning techniques, into smaller groups. Now, what you're going to find is that you throw away a lot of your proteins because not everything bins and things just kind of fall off. But what does fall into these bins or these categories, you can get taxonomy information. So you have function and taxonomy information for these about 300,000 proteins. The other thing we had for this system is this collection of isolates. And so we got 39 dominant bacterial isolates with fully sequenced genomes. And we have about 200,000 proteins from these isolates. Now, if I had my way and I had Star Trek type of technology, I would actually want to use isolates for everything, because you have discretely sequenced genomes that have functional annotations for it. So you have taxonomy, you have function, you have all your information. However, you can't isolate and sequence everything. So that's why we use these comprehensive, these comprehensive and binned metagenomes as well. So each of these are, again, you use each of these genomes depending on whether you can tolerate ambiguity, whether you want to know function, whether you want to know taxonomy, and honestly, what's available. A lot of times, the only thing you have is this comprehensive metagenome. So I think I went through this. The deep sequenced metagenome gives us broad functional data. The collection of bin genomes gives us focused taxonomic and functional data. And the collection of genomes gives us a very refined look at the taxonomy and it's not just taxonomy, it's species level identification as well as functional data. Now, if we start comparing total proteins, and I actually have 2 binned metagenomes. We have a binned from an all assembled metagenome, and then we also have a metagenome from the rhizosphere component that was also binned. So you can see that from the all assembled metagenome, again, that 30 million total proteins, if we bin those, we get about 4 million protein calls. If we do the rhizosphere, for some reason, I don't have the rhizosphere all metagenome on this slide, and I looked for it and I couldn't find it. Why am I doing that? I get about 200,000 total proteins, and there's about another 200,000 from the individual genomes. We get about 113,000 peptides from the all assembled genome, 69,000 from our binned deep metagenome, 48,000, 49,000 from our rhizosphere metagenome, and about 17,000 from our individual isolates. Now, this low number is telling me that there's not a lot of peptides that we're seeing. But then when we start going to proteins, and this is all done with a 1% FDR with 2 extractions that they had to be seen in 2 extractions and 2 analysis replicates and 2 peptides for protein. Now, I wanted to talk a little bit about unique peptides versus non-unique peptides, because unique peptides are peptides that belong to a particular protein. And when you're doing xenic cultures or mammalian cultures, plant cells, using unique peptides is great because there's not a lot of overlap. You know, if you have a plant, if you're working with a plant, you have one glycolytic cycle, you have one set of photosynthesis genes, you might have isoforms, but you only have one pathway. When you're working with metagenomes and you have even a simple 8, say you only have an 8-member consortia, you still have 8 TCA cycles, you have 8 glycolytic cycles, you have 8 amino acid synthesis cycles. And since in biochemistry, sequence belies structure, structure belies function, there's a lot of homogeneity between the proteins in these pathways. Therefore, peptides are not going to be unique. They're going to map to more than one protein because there's homology. So it's really difficult to use unique peptides in your metagenome analysis because you're going to lose so much information just because the homology is saying they should be the same. So none of this is with non-unique proteins. None of this is just with unique proteins. It's all non-unique. So we can see we get 66,000 proteins from our all assembled metagenome. We get 28,000 bind from the bind. We get 7,000 from a rhizosphere metagenome and about 6,000 from our isolates. Now you can say, wow, my gosh, 66,000 proteins, isn't that great? Well, is it? Because it's only about 0.2% of the total proteins possible. Whereas if you use a more refined metagenome, like your individual isolates, we actually get about 3% of the total. So it all depends on what metagenome you use, depends on the question you're asking. If I want to look at function, I'm going to look at all of my proteins. And when I identify 6 phosphofructose kinases, because it comes from all of the proteins, I lump them all together and I just look at pathways. If I start going down to individual genome isolates, I can start looking at what happens to those organisms, but there's only 39 out of probably 100 and something different organisms. So I'm blind to everything we don't have you're blind to everything that doesn't either in the individual isolates or in your bins. So if we look at overlap, we can see that there's the comprehensive database is our biggest. The bind, there's a lot of overlap here, but we still get some uniques from the bind as well as the isolates. Not really surprising because the isolates probably have peptides that in our comprehensive database just fall below that ionization level, where even if we're fractionating, we're still not getting to that low level. So we're still seeing a lot of peptides for the isolates that we're not seeing in the soils. So what do we see? If we use our global database, and what I do is I treat the community as a single organism. If I'm using a global database, I am not looking at, I'm not looking at what belongs to what taxa. So I basically, as I said, I collapse all of my similar proteins into one pathway. So it looks like a single organism. I use KEGG pathways to orient everything. So all of my glycolytic proteins, there might be 50 of them. They're all just a representative of one. I'm focused on function and I have the broadest number of potential proteins. And so in this collapsing, I took 66,000 proteins and I collapsed it into what I'm calling 2,200 functions. And what I see is a lot we see quorum sensing proteins, we see stress response proteins, we're seeing a lot of energy metabolism, glycolysis, TCA cycle, nucleotide biosynthesis, transcription, amino acid biosynthesis. And what it's telling me is that this is a microbial community with abundance of nutrients. Now, remember what I told you before, we just added glucose to this community. So we're adding a sugar. The fact that we're coming up with a result that said it has a lot of food means that we actually saw what we expected to see. If we had come up with, If we hadn't started seeing sugar transporters, I would have been concerned. If we had started seeing like stress response to nutrient limitation, again, I would have been concerned. Maybe not concerned if it had been a nitrogen or a phosphorus limitation, because as you add sugars to a biological system, it uses a lot of the nitrogen and you can deplete that from the soil. But yeah, there's an abundance of nutrients because we basically added food. Now, if we look at our bins, and this is just our global taxonomic bin, you can take your proteins and you can put it into different bins. So in this case, we just took it down to the taxa level, and we have the proteobacteria, actinobacteria, acetobacteria, a little bit of the planktomycetes, the veromicrobia, et cetera. And you can create a phylum level distribution of all your proteins. Now, this is just from our genome. This is saying this is what is possible. But if we take it and compare it to what is actual, it's actually quite interesting, because proteobacteria should have dominated, because there's more proteins in the proteobacteria than all the rest of them, according to our pie chart. But we don't see as many. We see fewer proteobacteria than in actinobacteria. So that means that even though the proteobacteria should have had a bigger component to our system, the actinobacteria actually did. And the acetobacteria is really a lot smaller. And then the pies for the rest of them are what we would expect. But it just means that in this case, when we add glucose, and I apologize for my formatting, when this moved over to my new template, not everything worked. Obviously, when we added glucose, it spurred responses in the actinobacteria actually responded to the glucose by synthesizing more proteins. But then if we look at our individual isolate genomes, you can see the blue lines here. These are our isolates across the bottom. The blue lines are the total proteins that we would expect in a rank, and this is just a rank abundance profile of the total number of proteins percentage. So if we took it as a metagenome, this is the total number of proteins for each one of them. And our top ones are Bradyrhizobium. I am really bad with Latin names. Sulorubrobacter and Blastococcus. And we can see that we get this blue rank abundance curve. But interestingly, when we do the same thing with the proteins we saw, we aren't seeing a lot. The Bradyrhizobium, Sulorubrobacter, and Blastococcus are not the dominant ones. I mean, they are dominant, but they aren't, but we see a lot of the low abundance organisms as, we see a lot of the proteins from the low abundance organisms. Now, what this is telling me is that even though a lot of the proteins come from, even though the Bradyrhizobium is high in abundance, it's probably not expressing all of its proteins at the same time. Whereas a lot of these guys maybe, and we're actually picking them up and seeing them. So, and all this was done by taking the exact same data and searching it against these different genomes. And in this case, we actually are using unique peptides, because if the peptides go across all of them, then we'd see straight lines. So the proteomics can actually reveal a lot about what's going on in a microbiome that the rank abundance profile won't. Again, if you looked at the rank abundance profile just by genomics, you'd expect a lot of these organisms, whereas we're seeing representation across the board. So just to conclude this part, I just wanted to stop and say, no one sample prep method works for every soil type. Test each method with your soil samples to determine what the best method is, and do this with the experimental design considerations. If you want to use the, okay, I'm gonna speed up. If you wanna use the MPlex because you want your metabolites and your proteins coming out of the same ones, then use that one. If not, look at other ones. Fractionation comes at an opportunity cost, but it will help you, it will help you increase the depth of coverage on your peptides in your system. Each type of metagenome provides a different type of data. Unbin metagenomes are great for function. Bin metagenomes are great for looking for taxonomy and what functions are in the taxon. So the next thing I'm going to talk about really quickly is some stable isotope probing experiments. So the reason we added glucose at the beginning is because we were going to add C13 labeled glucose in this part of the experiment. So we can add C13 labeled glucose to our system, go through the same process and get our mass spectra out of this. Now, notice there's no separate separation for an unlabeled to labeled, because what happens is in the mass spectrometer, we can take our spectra and compare it back for our peptide and compare it back to our theoretical spectra and look for this tailing of peptides. If we see the tailing of peptides, we know that it has incorporated a C13 label into the peptide, and we can label it as, we can mark it as labeled. What this allows is for us to do in situ labeling without any extra separations, which you would have to do if you were doing, say, CYP metagenomics. You actually have to do the density centrifugation, and then you just look at what is heavy. We don't have any of that. We just look directly into our system. So using the same sort of databases we did before, looking at the comprehensive database, we can see that a lot of our peptides are labeled. We get glycolytic proteins that are labeled. Our TCA cycle are labeled. We're labeling our pentose phosphate pathway. We have triacylglyceride biosynthesis labeled. Our carotenoid biosynthesis labeled. We're looking at fatty acid biosynthesis is labeled. Again, not really surprising. We added glucose. It's a sugar. I would have expected glycolysis. I would not have expected gluconeogenesis because we just added glucose. I'm expecting utilization of sugars. Fatty acids, it has an abundance of energy. It's taking it and putting it into fats for later. It's making its triacylglycerides. So it's doing the things I would expect for a very well-nourished system. Some of the other labeled pathways are amino acid biosynthesis, DNA synthesis, RNA synthesis. Our sugar transporters are labeled, again, not surprising. Elongation factors and transcription factors. This is a well-fed, well-growing system. So I just showed you the rank abundance profile. If we overlay our labeled proteins on top of that, we can see that some of these guys out here are picking up label faster than some of our high abundance ones. So in proportion to what we would expect, because remember, this is all normalized to the total number of proteins we saw. So these guys are actually active and picking up the glucose and utilizing it, which tells us a lot about who is doing what in our system. Now, interestingly, when we look at our top three abundant microbes, we see evidence of both glycolysis, which I told you we would expect. We also see evidence of gluconeogenesis in different organisms. So for some reason, our Streptomyces, these organisms are actually processing the glucose, whereas our Bradyrhizobium, our Sphingomonas, Caulobacter, some of our higher level organisms are actually creating their own glucose because we see gluconeogenesis. So we're seeing both pathways at the same time, which isn't really that uncommon in a soil system because these organisms are doing different things at different times, and soil is very diverse anyway. So you could be seeing different organisms doing different things spatially, which we don't have the ability to get to quite yet. So the isolates demonstrate that both pathways there are isolates to demonstrate both pathways, but this data represents the fact that we can see competing pathways in different organisms. So in our top three, we see, we see a lot of the same sort of glycolysis, TCA cycle, amino acid synthesis in our CYP results. So we're able to look and say what is actually active in our samples by using the CYP. So it's not just an overall protein, it's what was made after I added my label. So it tells me activity at a T0. Using a CYP gives you a T0. And then you can look what happens after that because you're only looking at what has actually picked up that label. So the last thing I want to, so just to conclude this really quickly, the stable isotope probeling provides temporal information because you can take time points. You have to take time points over to see where your label's going. It can identify active taxonomy, and it can also identify active functions in your microbial community. Now, the last thing I'm going to go through, I hope fairly quickly, is are real-time monitoring, which is not proteomics. And what we're really interested in is how does drought affect the CO2 release from our systems? And so we're looking at drought is becoming more common across the United States. It's been known for 50 years that when you dry a soil down and put water, CO2 is released, and it's released almost immediately. But no one has really understood why or where this CO2 is coming from. This was observed almost 60 years ago, but when no one knows what the molecular underpinnings of it are. And so we've developed a method to measure the CO2 release in real time. Now, the CO2 can come from 3 different places. It can come from abiotic effects. If there's calcium carbonate or other carbonate in your soils, it will be released by water, just by the water displacing that calcium to carbonate bond. It can come from exoenzyme activities, because drying causes an increase in dead microbial biomass, which then is rapidly decomposed into microorganisms, or it can come from intracellular metabolism, where microbes rapidly metabolize intracellular compounds. Now we know that organisms or microbes, as they dry, increase osmolytes, or their compounds. They synthesize compounds intracellularly so that they can withstand drying. It's so basically they don't lose water. It's to keep the water inside the cell. However, when they're rapidly wet, they need to get rid of these osmolytes or they're going to burst. So we are hypothesizing that the CO2 release is initially coming from this intracellular metabolism. Our field site is actually in Prosser, Washington, where it's about 30 minutes from the lab. And we actually have a field site where we're looking at the growing of tall wheatgrass in this system. And the reason we wanted to take this system is because, first of all, we live in a desert, so everything's dry. Second of all, we water. So we're doing this drought to water many, many times, even during a week, depending on how often you water. So what we've done is we've developed an apparatus that allows us to take a soil sample, add water, and directly take and push a gas through. And so the gas is coming out, going into a mass spectrometer. This is a GCMS, and we're actually able to get a real-time analysis. And so a sample output is shown here, where each one of these little dots is actually a time point, and we're measuring CO2 release. We are actually able to take a measurement every 2 seconds. So we're able to let it go for 24 hours. So the density of information is pretty large. And what we saw when we did our initial CO2 measurements is when we add water, we see the exact same response. And the response it's almost an immediate response that we add water, we see the CO2 come up, and it's reproducible. This is a triplicate, and you can see we're seeing the same time, almost the same timing and the same arcs. It's also proportional to how much water we add. So the more water we add, the more CO2 we actually see being released. We wanted to say, is it an abiotic effect? So we took some soil, we autoclaved it, added water, and we see a release of CO2. But if we overlay that on live soil, you can see that the difference in the CO2 release is pretty significant. So we know that this is coming from some sort of biotic event, but what is that biotic event? So what we did is we added glucose to our system. Yeah, glucose is a theme through this. And what we saw is an initial release, and then we saw a huge release. Now, glucose is the thing that is going to be picked up by, it was dissolved in the water, and it's going to be picked up by our microbes almost immediately. So it's going to mimic that extracellular carbon. But notice our initial release we still see initial release, and then we see a huge release. So we didn't know what was happening, where the glucose was coming from, or where the glucose came into this. So what we did is we added, so that's just, if we overlay the live soil, we see this. So we thought that was the glucose. But we did another experiment. Again, intracellular, extracellular compounds. Sometimes I hate animation. We did another experiment where we used C13 glucose, because in the mass spectrometer, we can differentiate between C13 CO2 and C12 CO2. So what we see is if the C12 carbon dioxide comes up first, and we see the C13 carbon dioxide, and let's see if I animated this. No. So what we're concluding here is that our initial release is due to intracellular compounds that the cells can get out right away. They're probably metabolizing all of their osmolytes to get rid of them quickly. And then it's picking up extracellular glucose later on. The nice thing about this is we can actually see, so this is our C12, we can see other gases. So we know that the C12 starts at 38 seconds. The C13 doesn't start for 56 seconds. But we can also look at water. So our water starts coming up at 47 seconds. Our O2 actually starts decreasing at about 67 seconds. And our nitrogen, our nitrogen starts decreasing at 73 seconds. So that's our CO2. And we can put this along a timeline to see what's actually happening. So what we think is once we add the glucose, we're seeing, of course, the degradation of the glucose to CO2 and H2O, which gives us a release of the water. It is the take up of the oxygen, a concomitant take up of the nitrogen. We don't know why that is yet. And then the release of the CO2. So we're actually seeing the release of the CO2 and H2O and the uptake of the oxygen. And this is all in the same experiment. We didn't have to run this 5 times. We just look at the masses of the gases that are coming out. So this is all in the exact same experiment, all over the same time frame. So basically here, what we've done is we've developed a method to evaluate the microbial mechanisms at a very fine temporal granularity. We can examine numerous gases at the same time. We can look at things like glycolysis and with the release of the CO2 and the O2. And in what we saw in our soils is we believe that what is happening is that the intracellular carbon is being metabolized first, and then it's accessing the extracellular carbon. And with that, I'm going to acknowledge the people that helped. The Hopland research team was led out of Mary Firestone's group with Owen Brody, Evan Starr, Katerina Zalina, Ulas Karaza, and Jennifer Petridge. All the RTMS work was done with our SFA, our soil microbiome SFA team led by Kirsten Hofmackel right now. None of this could have been done without our P&L overall mass spectrometry group. And this was funded by the Department of Energy through EMSL, as well as grants, as well as grants in our research team. And it was all performed at the Environmental Molecular Sciences Laboratory. This is just my plug for the EMSL. You can put user proposals in, where someone puts a user proposal in and they're able to access the kind of instrumentation that I talked about through here. And so with that, I will thank you for your attention, and I will take any questions. Sorry, I hope I didn't just fire hose you all with information.
That's great, Mary. Thank you. Let's open it up for questions. So I will pop one in. I appreciated the discussion on sample prep. This is an area that, so we operate a mass spectrometry core. We've got a lot of expertise, but extracting proteins from soil is quite challenging. You mentioned the impact of different soil types. Do you have any trends as to what approach you would use for different types of soil that you might be able to pass on? Any generalizations, or is it really a trial and error type approach?
At the moment, I think we're still at trial and error. If I was actually to write a grant on, well, there's a lot of things I'd write a grant on, but if I was actually being given a lot of money to do something, what I'd really love to do is create a cookbook of types, or what I'd like to do is try different extraction techniques on different soils with different clay contents, different sand contents, and see if there are those trends. But at the moment, we are, we're under the gun to actually, people care about the results rather than care about the correlations on the different sample types. So right now, I think that it's just trial and error. It's whatever works best for your soil. And I think even if I did give you some sort of insights and trends, they'd probably be wrong for whatever soil you came up with.
Other questions? I was going to ask another detailed question. You mentioned binning for taxonomy. And do you have a particular tool that you use for that step?
I will admit I didn't do that step. That came out of Evan Starr and Ulas Karol. I can't ever say his name. Karasov did that.
Okay.
I know there's a number of binning techniques. I think they use the tetramer, the tetrameric. It's a technique where you look at formers of the nucleic acids, and you look, you basically say this former, this, and look and see which one and how they closely align. And then once you get the nucleic acid usage collected, you look and see if you can find any of like the ribosomal genes to figure out what the taxa is. And I know there are different methods out there, but I usually use what people give me.
Other questions? If not, we will thank Mary. And I really appreciated that. Mary was great. And I'm sure as we start wading through some real samples, this will become all the more valuable for us. So I really appreciate the primer on there.
Yeah, and feel free to reach out if you have questions.
Great.
I probably will not be the one to answer it because we have a very talented scientist in our laboratory. Her name's Carrie Nakora, who probably has forgotten more about processing soil samples for proteomics. I mean, literally, that was her master's thesis. So I guess I can say she wrote the book.
Yeah.
And just has a lot of experience in doing this. So, if you're coming across questions, we've talked about writing another kind of not review article, but kind of a methods article that does collect some of our insights into this.
No, that would be fantastic. I mean, even I particularly found it interesting just looking at the picture of your biphase, what was it, the Mplex prep on real soil, and seeing that protein layer that frankly looks like soil.
Yes.
Was pretty striking.
It's messy. Soil is messy, and it takes time, it takes effort, it takes practice. It's not something that you're going to get right away, but you just have to keep at it. And eventually, a lot of it is trial and error, and a lot of it is what works in your lab with your methods and your hands. So even if you follow someone else's protocol, it might not still work the same. You just kind of have to try different things and be creative.
So I have a question along the lines of the sample prep. Are all of these, is this on intact soil, or is there kind of a pre-separation of the microbes?
So there is a method so when I said indirect, it can be either. We can do a pre-separation of the microbes, which is the indirect method, which actually for Hopland seems to work really well. And you can kind of wash the microbes off the soil. The problem is you're going to bias toward those microbes that are easily come off, and not the ones that are tightly bound. However, if you lyse in situ, which basically should eliminate the bias of what comes off easily, you're biasing against that which will lyse easily in the presence of a soil. And so we do a bead beating and a sonication step, as well as the presence of a detergent to help liberate the proteins. So it's either method, and it all depends on what works well. I think if you have a sandy soil, it's a lot easier to get the proteins off because there's not as much minerals for the proteins to bind to. When you start getting into clays and loams and other types of soil, you start getting a lot more attachment of the microbes onto the soil. And I just gave you a generalization that you can probably prove me wrong in whatever next soil you have.
Thank you.
Any other questions, everyone?
My slideshow.
Okay. Well, I think we will end here then. Thanks everybody for attending, and do keep in mind that we have some other seminars coming up over the next several weeks. Thanks everyone, and thanks again, Mary. I appreciated it.
And thanks for your attention, and thanks for the invitation. This has been fun.
Our pleasure.
And have a happy 4th of July.
You too.
All right, bye.
Bye.